First of all, it's the inspiration for my scientific work. The clinical care and the clinical issues that I see are what, inspire me to try to do the science. But it also informs what I think about the science.
I'm John Foxe, Director of the Monte Institute for Neuroscience here at the University of Rochester, and I'd like to welcome you to another episode of Neuroscience Perspectives. Please like, comment, follow anywhere you listen to your podcast if you like this content. I'm honoured to be joined today by the internationally recognised pediatric neurologist, Dr. Jeff Neul, the Annette Schaefer-Eskind Chair and Director at Vanderbilt Kennedy Center. Renowned for groundbreaking research into genetic neurodevelopmental disorders, Dr. Neul leads innovative studies on Rett syndrome, aiming to transform diagnostics and therapies for children with this rare condition. He's currently leading a landmark multi-site clinical trial that compares 3 FDA-approved drugs for Rett syndrome. Dr. Neul has received the Circle of Angels Award for his dedication and outstanding contributions to Rett syndrome research and is an elected member of the American Society for Clinical Investigation. Thank you all for joining us today. And Jeff, thank you for being here with us on Neuroscience Perspectives. It's really great to have you in Rochester.
Well, thanks a lot, John, for inviting me here. It's great to be here in Rochester. It's my first time. It's pretty nice to be here.
We'll have to show you around. Yeah. Perfect day. We ordered up the weather especially for you. Well, I've been really looking forward to having you in that chair there. You and I know each other from the IDDRC network, the Intellectual Developmental Disabilities Research Centres, and really for many years for working in biomarker development and rare neurodevelopmental disorders. So I want to dive into that. And the other thing about you, of course, is I know of you as a passionate advocate and somebody who doesn't hold back when he's talking about these things. So I think our audience are really going to enjoy that. Let's dive right into Rett syndrome. Help the listener understand Rett syndrome and why it's so key, why we spend so much time on that particular disease, why it's so important to you.
Yeah, Rett syndrome is a disorder that primarily affects girls, and it has a very characteristic pattern of disease where the children are born normally and have what appears to be apparently normal development for the first approximately 6 months of life. But then they start not gaining skills as quickly as they should, so they have a delay. But then about 18 to 30 months, they regress. And specifically, they lose acquired hand skills and they lose acquired spoken language. And they develop a characteristic repetitive hand movement and have trouble walking. And then they stabilize, but they don't really regain those skills. And then that's lifelong. And then later in life, they start having a late motor decline where they actually have Parkinsonian features, and slowness of movement. And I think that part of the reason that Rett syndrome, albeit a rare disease, although it's at the common end of the rare disorders, it manifests many clinical features that we see in other disorders. It was considered an autism spectrum disorder back 20 years ago because during the period of regression, which happens around the time when you might have an autistic regression, people might be socially withdrawn and be diagnosed initially with regression until they manifest with the clear hand stereotypies. And then additionally, as they get older and they start having these more Parkinsonian features, now you're starting to manifest that. So I think that's one of the reasons why there's maybe a lot of interest in this relatively rare disorder that has a genetic basis too. So 25 years ago, Huda-Zogby found the gene for Rett syndrome. And so that let us understand the disorder more, create disease models, and now really have a much broader sense of the phenotypic spectrum that people manifest. And in fact, with modern genetic testing, I'm starting to have people come to my clinic I would have never dreamed had a mutation in this gene.
Is that right? So well, let me just go back really quickly because I want to come to that. That what you're saying there is that there are people showing up with the mutation in the gene who don't have a full-blown Rett syndrome clinically. So we'll come to that. We're going to park that for a little bit because I also want to go back and pick up on something that you said there that I think is really crucial, which is that for a large chunk of, let's say, clinical history, a girl with Rett syndrome would have been diagnosed with autism. But in large part, that was because we didn't know of the causal gene mutation and how that's a key change, right? When you know, oh, there's a mutated gene, we can now put this into a completely different bucket.
Yeah, I mean, it was originally described in 1966. It really, but it was published in a Viennese medical newsletter, weekly newsletter in German, and so it really wasn't widely recognized until the early '80s, when Bengt Hagberg and colleagues published an English language case series in the journal Neurology,
Right.
and that really is what made people recognize this disorder about in the '80s.
Yeah.
But even then, there were a lot of confusion about what was the cause, because it really only occurred sporadically. It didn't occur in pedigrees and families.
Right,
now we
right.
know that's because it's what's called a de novo mutation. It's a new mutation that neither mom nor dad has.
It doesn't come down the family
tree. It looked like a sporadic thing. It looked like maybe it was an environmental exposure or an in utero insult or a mitochondrial disease. A lot of people thought it might be a mitochondrial disease that has unusual patterns of inheritance too. And it had these autistic features. So it really kind of perplexed people until the late 90s when the genetic basis was discovered.
And it really goes to the heart of why it's so important to be able to genotype as best we can, do deep genotyping on children with neurodevelopmental disabilities, because many of them are out there with undiagnosed syndrome, what we'd call a syndromic form, where we could in fact say, oh, this is what's going on here. And that changes the path of treatment and the path of investigation for scientists.
Yeah, definitely changes the path of investigation and allows us to group and parse and maybe stratify to develop different levels of understanding.
Right.
Hard to pick apart when you've got to add mixtures of different causes.
That's right.
Now, when you separate them out, you might get a cleaner signal.
Right.
So I think that's an important thing. And I think now we're entering an era that it is guiding potentially treatments.
Right. Because when you know that mutation, that opens a door to the fundamental molecular biology. Those mutations are in genes that are making proteins that are important for the building blocks of development, and knowing that is points or ground zero for understanding.
Absolutely, and it gives you the opportunity to use engineering to create disease models.
Right.
And that's really critical, I think, when we start thinking about things that translate. If we want to translate, we want to investigate therapies in a disease model or investigate biomarkers that we can translate between humans and animals or other disease models. That opens up a lot of opportunities to help accelerate clinical development of potential therapeutics.
Yeah. All right, let's come back to this business of folks showing up with a mutation that is, should cause Rett syndrome, but they've got a very mild phenotype. They're not that sick. They're not that compromised. There's a spectrum of severity. And where does that arise from? Try and help our viewers understand that.
People who meet the clinical criteria for Rett syndrome, 95% of them will have one of these pathogenic variants, these loss of function variants in this X-linked gene, methyl CpG binding protein 2, MECP2.
MECP2.
But we don't know the converse.
Right.
Because since the gene was discovered in 1999 till more recently, you had to have a clinical suspicion to do the genetic test, and you would genetically test that one gene.
Right.
Now, with the way genetic technology has advanced and the costs have come down, people are getting all the genes in their body sequenced, or all the DNA, all 3 trillion base pairs sequenced when they're suspected something is wrong. And so now we're starting to find people who don't fall into that classic bucket, right? And they are, first of all, as I said, it typically had been considered almost predominant, and it's still classic Rett syndrome is predominantly in girls. But now we're finding many more boys who have these same types of mutations. And classically, they were very severely affected right from birth and really didn't gain skills and oftentimes would die very early in life. But we've found now there's a much larger spectrum of people that who are boys who have these. In fact, people are coming out of the woodwork and coming to my clinic 20-some-year-old boys who walk and talk and drive and have whole spectrum of neuropsychiatric issues, right? And
So a different clinical manifestation.
yeah, and that might not be exactly a mutation that causes Rett syndrome in a girl, but it's now actually the pattern of it's in the family where mom may be unaffected or have a mild intellectual disability. The boys, her sons, and maybe the uncles, had a more severe manifestation.
Gotcha.
But I think it's really fascinating, some of these neuropsychiatric, extreme obsessive compulsive,
Interesting.
rigidity behaviors,
Well, now, so I'm learning something new here, and I thought I knew quite a lot about milder phenotypes and in some of the girls. So where we think of these nonverbal, very compromised girls in wheelchairs as a classic Rett syndrome, every now and again, a 15-year-old shows up that's walking and has some language, maybe not very, very complex language. And where the explanation for that is it the mutation against other genetic backgrounds?
Yeah, I mean, it's exactly, and something I'm very, very interested in. And yes, I mean, I used to follow a woman who was in high school, and she would walk in and tell me stories about her gym teacher.
Look at that.
Right. Yeah. And she could write and write her name, and she could do a spiral and had a tremor like someone with Parkinson's.
Right.
Now, those are exceptional cases, but she still had Rett syndrome.
Yeah.
Are
they though? Are they exceptional cases, or is it the case that other girls like her out there are all over the place and nobody's recognizing Rett syndrome because it doesn't look like a classic Rett syndrome?
It could be. I mean, it absolutely could be. You've really hit the nail right there. Is that I always say that people invert the positive predictive value.
Right.
There's a high positive predictive value if you have clinical Rett that you'll have a MECP2 mutation.
Right, 95%.
But we don't know.
Yeah. Because we haven't run that race at all.
The only thing we can go on are very large population genetic studies,
Right.
that are just UK Biobank or 23andMe or all of us, where they're just getting
hundreds of thousands,
hundreds
millions
of
of
people
genotypes.
who are, who don't have any overt clinical problem. They have a variety of typical population
stuff.
High blood pressure, these kind of things. But so that's the only thing we can really go on. But, I think there's probably a lot of, many more than we recognize. Because if we don't see the clinical features, we didn't do the test.
Yeah,
but, now with the testing being much more open-ended, you don't have to have the expert clinical prognosis to be able to do pretty open-ended genetic testing. Well, completely open-ended, basically.
I've asked this question already, actually, a couple of guests over the months and years. Should we be doing deep genotyping on every infant born in the United States?
Yeah, I think that I have mixed feelings about it.
Yeah.
I think that, for a long time, I remember when I first started learning about Rett, one of the things people said is that, Rett syndrome is more common than phenylketonuria. And phenylketonuria has been on the newborn screen for decades and decades and decades.
Yeah.
The difference is that with phenylketonuria, if you change the diet, you can change the neurodevelopmental outcome as an infant. And, without an intervention to change things, just knowing that your child has this mutation in a gene and has an increased risk of Rett with nothing to do potentially just creates undue burden. The flip side is that, well, what if there is things you could do?
Yeah.
What if you know that there's a risk and you can start intervening with therapies?
Right.
More intensive therapies. What if now when gene therapy trials are happening, what if they work? Should we be giving people gene therapy? However, the problem is it goes back to what I mentioned before about the positive predictive value. We don't really know of any random newborn who had a mutation in MECP2 what the likelihood is that they're going to go on to get Rett syndrome.
That's the piece that we don't really fully know now.
Don't know.
And maybe at one point we would have said if you've got a MECP2 mutation in a girl, the chances of getting Rett are very, very high. But we actually don't really fundamentally know that.
We actually don't know. But I think that's why it's critical, especially with the development of advanced therapies, which themselves carry not unrealistic and not insignificant risk.
Absolutely. Yeah.
That we have a better way to predict it. That, the mutation, I think, itself for a 1-month-old is not sufficient.
Yeah.
However, we know from work that I was involved in a natural history study, when we created growth charts specific for Rett, that actually the head size starts falling off the growth chart probably in the first 2 months of life. So with careful head measurements, and that's well before the delay or the regression. Maybe a combination of that, and plus other work by folks in Germany and Austria, looking at retrospective videos, home videos, and looking at subtle infant movements and infant vocalizations, and being able to look at, with the expert trainer, say this is different, and from the people who didn't go on to Rett. And then they've actually trained with machine learning to actually train models to recognize these vocalization pattern differences.
Yeah.
And so those are retrospective studies. It'd be great if we could do them prospectively, which, now, say here in New York, there is a, research, newborn screening, whole genome. And I know one of your former colleagues has, been involved in seeing some of these very young children who have been identified, and that provides an opportunity, I think. Right.
So it's, I liken it to, you send up a flare with the genetics. You don't get over your skis at this point, but you say, okay, this is a kid we need to watch. We need to watch the developmental trajectory. This kid has a mutation. They are somewhat likely to develop a disease that's going to really impact them in life. And by the time they're 6 months of age, we'll probably be able to tell with a much higher degree of certitude that this is going to happen, at which point we can really do something. And I don't think it's a tough concept for people to understand that early intervention is clearly good intervention. It very seldom does bad stuff. It's hard to imagine it doing bad stuff. Without that screening and without being in a major metropolitan center near Children's Boston or, the Kennedy Krieger Center and that, what is the age of first diagnosis for Rett syndrome? When are these kids picked up in the regular world?
Yeah, they're picked up about 2.5 to 3 years old, which is also, for many of them, after months to years of concern being raised by the family.
Showing up in ERs and things like that.
No, just be raising to their primary care physician, their pediatrician, maybe even to a specialist to say, my child's not developing, isn't gaining skills quite as well as their siblings or their friends or their cousins. And there's a lot of, watchful waiting, it's,
it might be okay.
Kids
go in different
things. But even then, after the point of sometimes some of the beginning of the regression, it might still take a while. Typically, though, the regression is the point that pushes things over.
So, but it says a lot, right? Because as you said before, you could get to a point of near certitude or very close to it that a kid is going to have Rett syndrome by the time they're 6 or 8 months of age. But the pickup time is 2.5 to 3 years. So that's at least 2 years.
Yeah, I think that it's potential. I'll just clarify. Within the first 6 months, it's always been said they're apparently normally developing.
Yeah.
And that's because generally they're meeting the general developmental milestones in the 6 months. The studies I was mentioning were much more nuanced and specific, looking at very specific, infant movements, subtle infant movements, subtle infant vocalization differences. So it's not usually until more the developmental milestones that occur after 6 months. But you can definitely see, and actually I've been looking at this very recently from the natural history study, the developmental milestone data, a large fraction of the children do not meet milestones, some of the major milestones by the CDC recommended guidelines, which is the 75th percentile, which is called act early, intervene, or something like that. CDC has a name, but and it's designed specifically for pediatricians and general health providers for screening to identify people. And a huge percentage of the children with Rett syndrome before their regression did not meet the milestones according to those guidelines, and,
Gotcha.
and should have gotten at least being referred to early intervention.
Right. Let's switch over to so you're running really important clinical trials in Rett syndrome right now with, I think, 3 different compounds. Can you tell us much about that at the moment? Is it?
Yeah. I mean, we've been trying to get that going. It's been
Gotcha.
a little bit of an adventure. Basically, it's 3 approved FDA compounds. But just because they're approved doesn't mean the FDA is keen on us giving them to children.
Fair enough.
So, and that's been a struggle from the regulatory side.
Gotcha. Okay.
So we previously did a small scale study of oral ketamine in Rett syndrome, and that's one of the ones we wanted to include in this trial. Giving oral ketamine for the longer period of time, 12 to 16 months, the FDA wanted more neurotoxicity, juvenile neurotoxicity studies. And the other drug is a histone deacetylase compound that's an anti-cancer drug. The drug is donepezil, which is used commonly in Alzheimer's. And for all of those 3 drugs, there's preclinical evidence of efficacy in disease models.
Right.
So
disease models specifically of Rett.
Of Rett syndrome.
So in animals with a Rett syndrome mutation who have a Rett syndrome phenotype, have a measurable outcome, these compounds have been used and shown to improve outcomes.
Yes.
So there's good reason to do this.
And it was animals in cell models, iPSC, organoid models,
Understood,
a variety of things.
Yeah. And of course, then the FDA, in fairness, stand as a guardian.
They're safety.
They're safety first, which
safety
is fair.
first,
right. But can be frustrating and
it
slow
can be
down
frustrating.
the process.
It's been a learning curve for me to learn about the nuances and challenges on the regulatory side. When you're scientists, you're thinking, oh, we're doing the best science.
Yeah.
But then when you start dealing in clinical development and with humans and regulatory issues, the safety becomes the top priority. Rightly so.
Absolutely. Yeah. Good checks and balances, really, right? Yeah. Are you hopeful with Rett syndrome? And I know these particular 3 compounds, but we also know that there are major gene editing therapy trials going on right now. When you put your finger in the air, what do you think?
Well, I think that it's it was exciting to get the first FDA approved drug for Rett syndrome a couple of years ago, and I think that now there's first of all that demonstrated that you could do a successful clinical trial and clinical development in Rett syndrome and in a neurodevelopmental disorder. Unfortunately, there's been a long list of failures, things that really, seemed like they had great scientific evidence and great preclinical evidence.
Right.
And so I think that was discouraging for people developing therapies.
Right.
And so it's good that shows that there is a way.
Yeah.
I think that there are a number of different avenues that people are exploring, like small repurposing molecules, like we're talking about, drugs that are already approved.
Yeah.
New development of small molecules that might be targeting specific biochemical, neurochemical systems that we know from the disease model are abnormal, but also then going right to the heart of the biology, either replacing the gene with gene therapy or correcting genetic deficits. And, we're in the point now where, 25 years ago when I got into Rett syndrome, it was always the dream of doing, a genetic disorder. One of the dreams is that there is a genetic way to treat it.
Yeah.
And so it's an exciting time that these things are being tested. Unfortunately, we are learning that they're dangerous, and we have to be careful and we have to be cautious. And with gene therapy, and not just in Rett syndrome, there have been a number of tragic events in Duchenne's muscular dystrophy and other neurodevelopmental disorders very recently. Right. And I think there's a lot for us to learn. And I think as neuroscientists, it's in a world of things like virology and immunology and immune response that maybe we haven't been paying as much attention to the science
right.
right?
Yeah. No, it's a very good point, right? Because you have neuroscientists thinking about neuronal function and this class of cells that inhabit the brain, but a big class of cells up there are neuroimmune cells, and we don't have the same level of understanding of immunology. Right.
Yeah. And for a lot of these, it just is a generalized. The problems become the virus and the viral vectors for the gene therapy, and just the immune response.
Right. It's interesting too. In some ways, we have these labels for ourselves. So I know I certainly think of myself as a neuroscientist, as I think you do. But as you're mentioning, we spend a lot of time now these days talking about immunology, and certainly genetics is a complete. I mean, we've had to completely retool around genetics. That was not part of, or not a significant part of my training originally.
It was for me. I was trained as a fruit fly geneticist.
Fair enough. Okay. So. Well, actually, let's do training because the other piece that's important, the distinction between you and I, is you're a doc as well. You actually could see a patient, and so you wear both hats. How does that play into your philosophy and the way you approach working in disease models as a practitioner, a clinician, and as a scientist?
It's a two-way information exchange is how I always think about it. I mean, I think that, first of all, it's the inspiration for my scientific work. The clinical care and the clinical issues that I see are what, inspire me to try to do the science. But it also informs where I want to what I think about the science. So, I mean, it informs, if I am in clinic and, seeing people with Rett syndrome and they have breathing abnormalities, I think, well, what can I study in the mouse? How can I measure the breathing and characterize the breathing and see if they have the breathing problems? And if they do, understand why these mice have the same manifestation of a clinical problem, these breathing abnormalities, and how can I use that? And then is there ways to develop ideas of how you do treatments? So, and I always describe my lab as being very much a translational lab. Translational, that's like a Rorschach test. It means different things to different anybody looks at. But the direction of my lab is almost always focused with the end in mind, and the end is doing something that's going to get to help clinical care. Now, it may be a ways off, right? And it may be somewhat indirect, but it may be, that it's
that's
guided
the guiding principle
by that, of thinking, what can we do that will develop a understanding of the pathophysiology? What can we do that will develop, a potential therapeutic idea? Or what can we do that will have a correlative biomarker between an animal model and a person?
Yeah. And does it ever lead to any conflict in you where you think, I need to be in the lab, I should be in the lab, or a piece of you going, the other piece of you going, I need to get into the clinic and treat my patients? Does that come up too? The flip side
and
of
then
it?
you need to sleep.
And you need to sleep too. I suspect not a lot of that goes on, but
it is true. But I think that I'm very passionate about moving the knowledge forward, both in the basic translational lab and in the clinical research space. And I think that there's a lot of reward and hopefully benefit to the people that I see as patients. But I do think that I might be able to make a bigger impact by doing clinical and basic research,
right?
Because there's only so many hours I can see one patient at a time,
right.
Whereas if we can develop something that would help lots and lots of patients, that's more impactful.
Often a little bit jealous of you fellas and ladies who have that clinical component. When I was a younger scientist, I used to go in the darkroom every now and again just to make some photographs, just so that I would have something to show for a day's labor, which is not how it works in science. There's very little that. It's such a long game, and it can be very tough on the feedback loop to, you're really playing. The work you're doing today could be 3, 4, 5, 6, 7 years down the road before a real product comes from it, if one comes at all.
Yeah, no, it's a good point. And that's exactly, it's interesting you raise that. I thought about that when I was doing training as an MD PhD and then doing residency training, is that there's a lot more immediate day-to-day gratification when you do direct clinical care.
You help somebody today.
When you're doing science and you're in the lab or doing things, you're months, years doing
Exactly,
until you get to the feedback and the results and the gratification.
So there's two things I want to go into with you. One is a bit about policy, but let's come back to that. Tell us a little bit about the young Jeff Neul, where the science bug came from, when you first realized this is my path. Sometimes I have people who are here and it thinks the first thing I ever thought, the first time I dressed up for Halloween, I had a doctor's coat on.
No. I actually went to college at the University of Illinois, and I started in chemical engineering.
Wow.
And because I like engineering, I like physics, I like chemistry. And I got into the chemical engineering classes in my second year, and I realized chemical engineers didn't do what I thought they did. I thought we were going to engineer chemicals. Actually, nowadays they kind of do, but I thought we were going to engineer chemicals, and we were just mostly moving fluid from one place to the other at a certain rate and losing heat along the way.
I see.
Which was fun, but and it turned out organic chemistry, synthetic chemistry was really more what I had envisioned chemical engineering to be. And so I switched to chemistry, and I had just thought I was going to go and get a PhD in chemistry, like physical chemistry or physical organic chemistry. I started getting interested in the fact that on the periodic table, sodium and lithium are right next to each other. And we eat sodium chloride all the time. It's a vital element,
salt
for all of the homeostatic processes in our body. But it doesn't have any obvious neuropsychiatric effects, whereas lithium people use for bipolar and mania.
Yeah.
And the aspect of those things from a chemist standpoint was really fascinating to me. How their general chemical structures are very, very similar.
How interesting.
And so I was really fascinated by that. And so I got interested in the brain and neuroscience from a chemical perspective from a neuropsychiatric perspective. And I was doing an internship at a pharmaceutical company that no longer exists near my house because it was bought out by other people. But and I was doing organic chemistry for the summer, and being kind of a brash 19, 20-year-old, I was flipping through.
It's a good thing you've calmed down.
This is back in the day when there was books of the telephone listing, and I was flipping through the book, and I saw this guy who was the VP of research had an MD PhD, and I was like, why would anybody have an MD PhD? So I picked up the phone and dialed it and got his, admin and admin assistant, and she just put me on the schedule. And so 2 weeks later, I walked and sat down, and he just looked at me like,
who are you?
Why are you in my office?
I love it.
But he had explained to me, he was a microbiologist, but he explained to me about, I told him my interests and exactly what I told you. And he said, well, obviously you can get a PhD in neuroscience or chemistry. He's like, but if you ever want to do human research and have human subjects, if you get the PhD, you're going to be a scientist, but you get the MD, you can understand the clinical problems and do it. And that's when I decided. So it wasn't until my junior year I decided to go to medicine.
So literally, a random guy you picked out of the phone book who had these.
Well, he was the VP of research.
Yeah, fair enough. But you went through the phone book to get to him.
Yeah,
and he changed your life.
Yeah, he changed my life. Yeah,
amazing.
That was one change in my life. The second change in my life was when I was finishing medical school, my fourth year in medical school, and I knew I was going to go into child neurology, and I did my MD PhD at the University of Chicago, and I was doing a reading course with a senior child neurologist there who was very influential for me, named Peter Huttenlocher, on the neurobiology of autism. Mm-hmm. Peter Huttenlocher is very well known for doing all this human neuroanatomy studies in terms of the exuberant synaptogenesis and pruning, and the pruning that occurs over the lifespan in the brain, starting at the base, going up to the forebrain, not even pruning all the way out until like late adolescence to the prefrontal cortex. But it was October of 1999, and I asked him, where should I train for child neurology? And he said, oh, Boston Children's Hospital. And he said, but and he turned, and on his desk, open, there was a journal. And he picked it up, and he said, Huda Zogby found the gene for Rett syndrome. You should go work for her. And it was October 99. That's when they came out.
Wow.
I think probably I'd asked him a week earlier.
It wouldn't have happened.
I think he was reading that article that day.
Life is such a journey, and you're a feather in the wind sometimes. Yeah. With the true purpose though, because you were heading down this path. This was the path.
Yes.
Wow. And what exceptional happenstance to end up with
I know.
The grand maven of Rett syndrome.
Yeah. I didn't really know much about Rett syndrome. I barely knew it. I was interested in autism and neurobiology of autism.
Yeah.
I didn't really know about Rett syndrome. I didn't know much else.
And just going back then, I don't know how many 10-year-olds there are out there going, I want to be a chemical engineer. Maybe there are. Where did that come from? Was it the household? Was it?
Bless my parents. When I was, probably in third, fourth grade, you write some sort of essay about what you want to be when you grow up. And I wanted to be a poet. And my mother taught English at the community college, and she introduced me to a local poet, and I interviewed him and wrote up a whole essay. And so I actually wanted to be a writer for a long time. And in high school, I did journalism, and I was really into the high school newspaper. And I was debating between going into some sort of engineering science or going to journalism.
Very good. And I know you're a gifted writer too, so that's always very helpful in communicating science.
Communication is a vital part of science.
It's a huge component.
Underappreciated.
Absolutely. And you are a great communicator. I know that from long experience with you. And it brings me to maybe the last thing we'll try and talk about, a little bit of policy, because again, you and I serve in leadership roles as part of the IDDRC network, and I don't think anybody would ever describe Jeff Neul as a shrinking violet, for example. You are an outspoken advocate for folks. You have strong opinions about it. When you look at the current moment we're in and you think about NIH funding and where the scientific enterprise is going, what do you think? I see it in your facial expression right now.
Of course I'm concerned. I think, after World War II, the investment the U.S. government made into science, both through the National Institutes of Health for health sciences, but also through the NSF, through the Department of Defense, all of these things, creating the aerospace industry, creating all the medical industry and medical advances. That's really what put this country where it is, and really the educational systems and supports that it fostered, that made us the envy of the world. Right. And it would be a real shame to see that go away. Right. I think that it's a great country, that we've done great things, and we have so much ability to continue doing great things, and really making a difference in people's lives. And I think for intellectual and developmental disabilities, it would seem that it would be an easy sell, because these are children, right? And they're people who really are deserving of our help, right? And trying to improve their lives and their families and their communities, right? And so I think that I would hope that that's an easy sell. Yeah.
And you're going to keep pushing hard to sell it. I think the mark of a great society is how it treats its most vulnerable citizens, and I think that's what you just expressed really beautifully. Jeff Neul, such a pleasure to have you here at Neuroscience Perspectives. Thanks for taking the time with us.
Well, thanks again, John, for having me here. I enjoyed my time.
Awesome.